All right. Has anyone picked up their lab packets yet?
Yeah.
Okay, cool. Um, so we're going to get a chance to use some of that pretty soon, as long as you've got your bread molding nicely. All right. I want to talk about your moldy bread real quickly. We may find a variety of fungi on there, but most likely, probably 99% of cases, what we're going to find from soil on bread are zygomycetes, and in particular, a group called Mucor. And so if you've read your chapters yet, you'll have seen that, but we'll get into it. It's one of the most common fungi on Earth because it tends to live in soil. It grows really fast. It's just highly abundant. And so these Mucor fungi, probably what you're going to find. And so whose bread is doing anything moldy looking?
Mine is. It's disgusting.
Okay, good. Is it green mold or black
Yes,
mold?
green.
I got a little bit of both.
Okay.
I have lots of green taken over.
Okay. Green is going to be ascomycete, so I'm hoping for black mold.
Okay.
You found some weird dirt where the ascomycetes are more abundant, but that's okay. I'm sure there's some black stuff on there.
Yeah, there's a little bit.
Yeah. So the black stuff is what we're going for pictures for that assignment. Okay.
What if it's more like of a whitish color?
Whitish is good.
That's
Yeah.
what mine is too. Okay.
So it'll be like white fuzz, and as it gets older, it's going to start getting little black spots on it. Okay.
Dr.
How
Zahn?
long do you want us to keep a hold of those?
Oh, keep it around. I mean, things changed for the better over the course of like a month or two. Yeah, when you become a mycologist, you just like find nasty stuff in the back of your fridge and you keep it, much to the chagrin of your family. I had an experiment where I just threw some oranges deep into our basement in a plastic bag with some water in there and just like waited. My wife was not happy about that, but it gets super fun. Let me see if I can find a picture here. All right. So I'm going to share my screen real quick. Okay. So essentially, this is kind of a little close-up. But this is what we're going for. You can see like the majority of the fungus is going to be white, but you'll start seeing little black dots. Those are these spore-bearing structures, these sporangia, okay? Largely what you will see with zygomycete mold on bread is white fuzz. It doesn't start turning black until it's like run out of food, basically. Okay, so here's another nice little image. All right, so you'll see white fuzz, you may have some green spots, but zygomycetes tend to grow faster than almost any other fungus out there in terms of just converting food to biomass. And so typically that's what's going to take over your bread is some blackening, darkening fuzz. All right. What I like to see pictures of for that, I want you to look for the hyphae and sporangia. Okay. Questions about what those terms are? You good on that?
I do have a related question.
Great.
So if it's been like 5 days and I'm not seeing any growth, should I go get some more dirt and
5 days?
Yeah.
I'd make a little more mud. There's a chance if you take dirt from like the surface, kind of like the exposed to sun, a lot of times the sun may have killed off stuff. So get down there into like some garden compost or, a few centimeters deep or whatever, and sprinkle some of that on there.
Okay.
Yeah.
Shouldn't you keep it like in a dark place instead of with sunlight? Would that help?
It actually likes diffuse light, and we'll see why today, but that helps it grow and start fruiting. So you probably are going to have a more difficult time getting the sporangia to start forming if it's in total darkness. Now, it doesn't need much light. If you see in the chapter, the light from one single star is enough to initiate fruiting. And so just like a few photons is really all it takes. But complete darkness is going to be tough for it.
Okay.
And this kind of makes sense ecologically, if you think about it. What would be the point of producing spores underground? They're not going to travel very far. Bugs aren't going to pick them up. And so it's got this mechanism to kind of help let it know that it's out of the dirt, it's exposed. And so kind of only then does it want to start making spores, or else it's kind of wasting its time. And so it needs a little bit of light. I think, throw it in your sock drawer or whatever, if you want your socks to smell awesome, and just open it once a day for a few seconds. That's going to be enough light. But don't throw it in aluminum foil into total darkness. All right. So in your lab packet, you got a couple single little dishes, one a big dish, and then a little tiny dish that's sealed up, and then you've got a stack of tubes. Okay. What you're about to do is try to transfer some of these zygomycetes onto one of those tubes in the stack. All right. What that is a nutrient auger. So we've added malt extract, yeast extract, some buffer, potassium phosphate, and then a small amount of antibacterial stuff in there. So like just your standard kind of penicillium, that kind of thing. So just a nice little mixture to kill off bacteria, because we don't want, this is mycology class, we don't want to grow bacteria and they're everywhere. All right. So how do you isolate bread mold onto a petri dish? Any ideas?
Real quick, you want us to transfer that bread mold to one of those many petri dishes that's in the long tube?
In the tube, yeah.
Okay.
So take that long tube, pull out one of those, keep the lid closed, then seal the tube back up with some tape. And we're going to try to get some zygomycetes to grow in more or less a pure culture. It's going to be tough without, a modern lab and sterile hood and everything, but we can do a pretty good shot at it. So how would you isolate that?
Is it kind of like how we do with bacteria, like streaking, or no?
Yeah, it can be that simple. What I would do is just take a pair of tweezers and hold them over a lighter to kind of like kill off all the bacteria that's on there already.
Mm-hmm.
Let them cool for a bit, and then just pinch a little bit of that white fuzz off of your bread. Or if you've got black spots, definitely get some of those black spots, and then just dab it into the center of that plate, put the lid back on, and then seal it up with tape or whatever. Okay. And that's it. Within probably overnight at that point, that's going to grow into a huge culture. Okay. And it'll probably be easier to visualize with your microscope that way, because you're not dealing with a bunch of bread particles. So the techniques of growing fungi in mycology are actually pretty straightforward, pretty low tech. And it's kind of a shock to people when they get into a mycology lab and it's just people walking around with tweezers moving bits of mold from one plate to another, but that's really all it takes, especially for these that are really easy to grow. Okay. So I want you to get on that because we're going to basically be building a fungal culture collection, and this will be your zygomycete, probably a mucor that you'll get growing. All right. So label that with a Sharpie, just the date and where it came from. So moldy bread. There's a fly attacking me, so if you see me waving, that's what's happening. All right. And so yeah, when you're looking at those under the scope and for your assignment, try to get photos of sporangia if you have them, and definitely the hyphae. All right.
Hey, Professor.
Yes.
So did you say that we just need like a little pinch of the mold in the petri dish?
Real tiny. Mm-hmm.
All right, cool. Thank you.
If you can barely see it, it's more than enough.
Okay.
Because all it's going to take is one spore to take over that petri dish, and that's microscopic, so it really doesn't take much.
And is that mold coming from our bread?
Yeah.
Okay.
Mm-hmm. So just find a spot of interest on your bread, pinch a little piece off, stick it into the middle of one of those plates, seal it up. And that'll store for up to a year without needing to be transferred or anything. Just room temperature, it'll be fine. Right, and so then you'll have a zygomycete. Now you don't want to like take that dish once it's all grown and got fuzzy and open it next to a bunch of fresh fruit that you just bought. So keep it sealed, or it will go through your house looking for food and eating it. All right. Questions? Anything so far? All right. Are we ready to talk about zygomycetes?
Yeah, let's do it.
Let's do it. All right. So zygomycetes is our next group of fungi. They are also paraphyletic. That's kind of the rule in fungal groups. We'll look at how they grow. We'll use them as an example for like Hyphal organization, how cell walls are laid down because they're nice and simple. We'll look at spores and how spores are formed, the different ways of doing that. Then we'll learn some more terminology, and we'll look at a couple groups, the mucors and the halobolus group, right? So on your dish or on your bread, we're basically looking for this image on the left, these asexual spores. Zygomycetes, though, as a group, have been grouped together morphologically, of course, by this structure, this sexual spore that we call a zygospore, hence zygomycetes. Okay, so anything that makes one of these has been traditionally grouped as a zygomycete. All right. So first we want to talk about hyphae, because we really haven't seen them to a great extent yet. When we looked at chytrids, they have these kind of rhizomorphs, you know, these hyphal-like structures, but not true hyphae in the sense that we tend to think of it. All of these images are not fungi. Right, so you look around and it looks like fungal stuff, but we've got oomycetes on here, we've got cable bacteria, we've got some weird amoeboid forms, so foraminifera. None of these are fungi, but they all have filamentous growth. Okay, so takeaway here is that filamentous growth has evolved multiple times across the tree of life, bacteria and eukaryotes. Okay. This is because it's a really good way to grow if you are a lysotroph. Can somebody refresh my memory? What is a lysotroph?
Well, troph is going to be for what it consumes or eats. Lice, I'm not sure.
Isn't it a breakdown, basically something that breaks down other components we eat?
Yeah, it breaks stuff down, and specifically lysotrophs break it down outside of their cells.
Oh, okay, cool.
So
they're
decomposers.
so they're excreting enzymes outside of their cell. It does the digestion out in the environment, and then it reabsorbs the digested stuff that it can actually make use of. Okay. Humans are actually lysotrophs also. We do all of our digestion outside of our cells. We just do it, in our stomach and intestines and whatnot. So we're excreting enzymes into the environment, but we're a tube shape. So we surround a piece of the environment exterior to our body, and we put food into it, excrete enzymes into our digestive system. It breaks down our food, and then we reabsorb the digested stuff. Okay, so opisthecons are almost all that. I mean, the corals are kind of the exception because they actually take up little food particles into their cell, and then inside the cell, that little food vacuole, they put enzymes into that, and it breaks it down, and then it's released into the cell. So that's called a phagotroph because they ingest their food into their cells. Fungi and bacteria are lysotrophs, okay, just like animals. So they're excreting enzymes into their environment, and this is potentially pretty inefficient when you think about it, because because those digestive enzymes are expensive to make. They take a lot of resources, and you've got some complex food. It's not edible yet. You've got to break it down. So you're next to it in the environment. You excrete some enzymes, it digests it. What can happen before you get to reabsorb those nutrients?
You'll lose a lot of them.
Yeah, somebody else comes along and takes them, right? And so there's like fierce competition. In fact, there are a lot of bacteria that ride around on fungal hyphae because they know that the fungi are excreting these enzymes. And so these bacteria are just like there to steal the digested food, and they don't even make their own digestive enzymes anymore. They just rely on fungi to do it for them. They're little thieves, okay? So if you're being a lysotroph, it's kind of tough. You have to morphologically adapt for the fact that you're excreting your enzymes. So there's a few ways to do that. You can have a really high surface to volume ratio. Okay, so you're really small, spherical shape, right, shown here. So a lot of fungi are yeasts, so like bread yeast. They get around this this efficiency problem by just being really tiny and proliferating, and so anywhere they are, they have a lot of surface area to uptake their digested stuff. Okay, animals have gone aside from corals and made a little tube shape, so we surround the environment, excrete enzymes into our digestive system, and then they have nowhere to go. Once those food particles are digested, we have access to all of them. Okay, but largely a lot of groups have kind of converged evolutionarily on this filament shape, and that's because it's got a really high surface to volume ratio, little tiny filaments, and it can kind of surround stuff also. So you look at this shape here in the top left. If this hypha excretes enzymes out to this side and digests some food, yeah, it may get to reabsorb some of those, but maybe some of them diffuse over here, and boom, they're captured by that hypha, part of the same body. Okay, so being a filament kind of lets you surround things also. So it's a handy shape to be. All right. Here you can see kind of a 3D structure of some hyphae growing in the top right, and you can really see how this surrounding works out in practice, right? If that's all the same organism, it kind of doesn't matter which hypha is the one that uptakes those nutrients. Somebody's going to get it. Okay, so that's really handy. Here on the left are also some hyphae, right? These rhizoids, but they have differentiated structure and kind of like a casing that tends to be darkened with melanin. Over here, this is the bread mold that you're looking at. It's a bunch of white fuzz. It's growing all through the bread. It's digesting those complex carbohydrates into simple sugars, and it's reabsorbing them. Okay. Hyphae can take a lot of different forms. When you look at a mushroom, that's just a bunch of hyphae all stuck together with some differentiation. We've got these weird rhizomorph forms here. We've got ectomycorrhizae, which congregate around the roots of certain plants. Okay. But hyphae, the point here is that hyphae aren't just simple filaments. When they get together, they can form complex structures and they can even differentiate. Okay. Questions so far about hyphae? All right.
Just a clarifying question.
Sure.
So hyphae, that's what extends when it's growing, correct?
Yes.
Okay, perfect.
Yeah, so the singular is hypha, and that's just a single little tube. So typically when a spore germinates, it starts growing a single tube. Hyphae is once it starts branching out and there's a mass of them. Then we have a mass that you can see with your naked eye. We call that, I'm brain farting here, mycelium, sorry. So a mycelium is just a mass of hyphae. It's kind of like the true body of the fungus. So when you look at your moldy bread, you're looking at the mycelium of the fungus, which is just a bunch of hyphae. Yeah.
Sorry. When you said that they differentiate, what do you mean? You mean like different functions? They acquire different functions? Or?
Yeah, different functions and then even different morphology. So in the top left here, you can see these look like roots, like black roots. That's actually masses of hyphae that have kind of grouped together. And the ones on the outside have differentiated and started laying down like a thicker chitin structure and then expressing melanin.
Oh, wow. Okay.
And in the middle are just a bunch of normal hyphae formed together like a little cable. And so that's cellular differentiation, way different than animals do.
Right.
We don't have the cadherins and adherins and stuff that animals do to be multicellular. Here we've just got close association and differentiation.
Thank you.
Yeah. All right. We can group our hyphae into two major types. All right. In the dikarya, the ascomycetes and basidiomycetes that we'll see shortly, we have septate hyphae. So they've got these little walls that kind of divide up each hypha into sections. Okay. You can see those, the arrows pointing to the septa. Okay. In chytrids and zygomycetes, they're aseptate, also known as cenocytic. Okay. So they don't have those septae in the main branching hyphae. You can see that it's all just kind of one cell, in essence. All right, pros and cons for these. What do you think?
The aseptate would allow exchanging nutrients easier.
Yes, absolutely. So it is a giant cell, in essence, and so nutrients can flow through it, kind of like a circulatory system. If you look at it under a microscope and have the patients sit there for a minute, you can actually see stuff streaming through the entire fruiting body. What's a benefit for having septa?
Would it be gradients are easier to form?
Oh yeah, so you could form easier gradients, and it helps with differentiation, for sure.
Could it also be protection? So, for example, if part of the body gets damaged, then it doesn't lose all the nutrients or whatever it has in there.
Absolutely. And so these are kind of like, the Titanic runs into the iceberg and they're like, hey, seal off these doors.
Yep. Mm-hmm.
That's what's actually easier to do in the dikarya with the septate hyphae, is that you can damage the end of a hypha and it's able to kind of quarantine and just break off at that point, and it doesn't kill the whole strand. Okay. So there's pros and cons for each of these, but we've seen that zygomycetes and chytridiomycetes are actually paraphyletic. They're just kind of based on some morphological features, but they do all seem to have aseptate hyphae. Okay, and so we think that that was the original condition of fungi, was that when they made hyphae, it was aseptate. It was just single-celled. And that's actually kind of matches the case for some of these other groups that we see. Cable bacteria being an exception there, but they're bacteria, they're weird, nobody cares. But for like oomycetes and stuff, they're also aseptate. Okay, so it's kind of seen across the eukaryotic tree. It's aseptate, and septate hyphae being kind of a recent innovation of fungi. Okay, but zygos don't have no septa. Every now and then they will lay down a little cross wall and segregate off a little piece of themselves. Okay. The main time they're doing this is for reproduction. All right. So for making spores, for example, they got to lay down cell walls between those spores, or else it's all just one big cell. All right. So here we've got kind of septa development in a zygomycete when it's making this sporangium. Okay, so this is that kind of fruiting body. It's asexual. What they do is they've got a little hyphae growing. It'll start swelling at the tip, kind of growing into a big bulbous ball, and then a bunch of nuclei move into it. So thousands and thousands of nuclei just kind of stream up into it from the rest of the fungal body, and then it blocks that off. So it makes a little septa right there. So you've got thousands of nuclei in the swollen little ball at the tip of a hypha, and then those nuclei start getting walls formed around them, right? So they go from nuclei here, they start growing, little walls form around them. That's just a bunch of circular septa. Basically, it's actually the same genes that get turned on as when making regular septa. And so suddenly you've got spores. So the upshot of this is that in a fungus, almost any part of the fungal body can become a spore, especially an asexual spore. All you got to do is put a septa there and let it break off, and suddenly you've got a spore. Okay. Some fungi are even they even do that as kind of their main reproductive strategy. We don't know that they have sex or they've lost the ability to do sex. We haven't seen it. All we know is that they grow as hyphae, and then suddenly they lay down solid cell walls, and bits of those hyphae break off. They get carried away by insects or washed away in the rain and start growing into a new hypha, a little clone. Okay. So we've got a bunch of different types of spores, and this just is annoying mycological terminology, but it follows a nice little pattern. Sporangiospores, produced in a sporangium. Okay, so that's most zygomycetes that we see are going to have sporangia. And what that is a swollen region that makes a bunch of spores. Okay, we got zygospores. It's produced by a zygosporangium. So this is the sexual reproductive in zygomycetes. Conidiospores produced on conidia. Okay. Conidium is just a fancy way of saying a spot that's making reproductive asexual spores. So over here, this is penicillium. So the famous fungus that has saved millions of lives since penicillin was put into mass production. Okay. So this is an ascomycete, but it's the best picture I could find of a nice line drawing of conidiospores. You can see the hypha growing here, and then it starts differentiating, and just little bits of it are segmented off, and suddenly that's a spore. And so we call it a conidiospore because the people who name these things have to have a special word for every possible structure. Okay. So conidiospores are formed on conidia. Sporangiospores are formed in sporangia, and so on. Okay. Questions?
I have a question.
Yes.
Do you know biochemically, I don't know, has any research been done on like how does the cell know that it needs to grow the cell wall to isolate it? Or do we know how like the cells communicate or?
So it's a combination, like everything, combination of genetics and environment. And most of the research has been done on environmental cues for kind of what triggers that cascade to lay down a cell wall. Starvation is a known one. So if they start running out of food, they will do this. Light is another one. And CO2 gradients seem to be pretty important. So if you get a sharp drop in CO2, that's a trigger also. All of these kind of go back to that same thing that you were talking about earlier with the light. Like if your main body is underground, which with fungi they are, or like in your bread, okay, they're growing through substrates, digesting it as they go, proliferating. It doesn't make sense for them to make spores in that condition. They need to be outside of their substrate. And so if they're growing through and they suddenly hit the air, that's going to be a sharp drop in CO2. And so that's a good trigger. Light is also a good trigger. And starvation, if you're running out of food, it's basically time to do sexual reproduction. That answer?
Yeah, great.
Okay.
Thank you.
As far as the genetics go, they're more complicated and work is ongoing on that, particularly with like plant diseases, because they cause billions of dollars of economic damage around the world every year. And so that's kind of where the research funding has gone for the genetics of fungal reproduction, is for agricultural diseases. Outside of those, very little is known because who's going to pay for it? So that's science for you. Okay. So in zygomycetes, we've got our vegetative stage, aseptate hyphae. We've got asexual reproduction in sporangiospores that we just saw right over here on the left. Okay. And we've got sexual reproduction, which we haven't really seen yet. And this is the zygospore. This is why they have their name. It's not because of phylogenetics. It's because they had this structure. We just call them a zygomycete. Okay. So zygospores is the sexual reproductive mode. And so that's the top image here. Okay. So we've got our hypha growing. It's haploid. Okay, so it's got one mating type. Another hypha will grow up and meet it. We call these progammetangia, just meaning that they're the things that are about to hold the gametes. All right. As they grow towards each other, when they touch, they send signals that say, hey, lay down a cell wall. And so they make a septum. All right. So here's the septum being formed. We've got a macrogammetangium and a microgammetangium. Now remember, gammetangium just means the thing that holds the gamete. All right. Okay, which one is male, which one is female?
Female's always the bigger one.
Yep, so the macrogammetangium, that's the female. So the big side gets a donation of a nucleus from the small side. So it goes that direction. So the microgammetangium sends in a haploid nucleus. Okay, they undergo karyogamy in there, and one of the nuclei that fuse and make it to diploid will undergo meiosis. The rest kind of die off, and then you've got your zygospore forming. Okay, pictured here, this is showing what you'll see in your textbook called a homothallic zygomycete, meaning that it can mate with itself. This is actually quite rare. I think there may be like 6 species that we know of, but it just makes for a prettier drawing. Most zygomycetes require a different fruiting or a different haploid body that has a compatible mating type to kind of grow into it. Okay. So keep in mind, this image is a good one showing the development of the gametangia and everything, but it's atypical in that this is one that can mate with itself. Typically, you got to have two different strains come together and find each other. All right. So this is showing we got our sexual reproduction and our asexual reproduction down here. All right. Here's a better look at some of the sexual structures, these zygospores, particularly here on the right side. So this is a light micrograph, and you can see the gametangia on each side of this, and then suspended in the middle is this sexual zygospore. Okay. So we got all our mycelium growing. If two compatible mating types meet up and they're ready to do sex, like they're running out of food or the weather is changing, it's getting colder and something like that, they say, hey, conditions are bad, it's time to roll the dice again with our genomics. Okay, so they come together. The microgametangium sends in some nuclei. They find the macrogametangium nuclei, and we get a zygospore. Okay. So this little drawing here at the bottom kind of shows the development of that. Now, I don't necessarily expect that you'll see this on your bread for a number of reasons. What might those be?
Favorable conditions.
What do you mean?
Like there's no reason for them to go through this process. Is that what you're?
Yeah, I don't think that on our time frame there's going to be any reason for them to do this. The piece of bread you've given them is probably somewhat large. That's a lot of food. Okay. There probably is plenty of CO2 in there, maybe even repressing some of their sporangium development. So if you can't get sporangia, maybe just open your bag a little, let some air in, and then seal it back up. That may induce formation of your sporangia. So there's plenty of food. They still think they're underground, and there's one more reason why this might not work on your bread. The odds are that you don't have homothallic species, because there's 1,000 times more heterothallic species. So what's that mean?
You need to have multiple individuals meet each other.
Mm-hmm. If you put down some dirt on bread, there's a decent chance that you've got compatible mating types in there, but probably only one of them is going to be best suited for your bread, and it's going to outcompete the others. Okay, so the odds are if that in any given little spot on your bread with the black mold on it, it's either going to be all male or all female. Okay, and the odds are also that it's not going to be able to mate with itself. It'll be heterothallic. And so how would you go about setting up conditions so that you did get these zygospores on your bread? So if I gave you an assignment, I want you to create and find some zygospores on bread. What might you do?
Would you get another piece of bread, grow some stuff on it, hope that it's the opposite, and then put them together?
Yeah, it's really that simple. Either that, or you take your piece of bread and on it you put some dirt on one part and dirt from another source on the other part, and wait for them to grow together to kind of meet up in the middle. Okay, so if you do that, the odds are much better that you're going to see zygospores, and they'll actually be right along the region where the two mycelia met up. Okay, they won't be on the outside, they won't be in the middle of where you put the dirt. It's just going to be like where the hyphae met each other. And you can actually do this. It doesn't always work every time, and so that's why I didn't bother having you do it for your assignment, but it's worth a shot to take two different sources, like a good meter apart, of dirt, and stick them on each end of your bread and wait for them to grow together. And a lot of times you'll get just like this line of really thick black dots. Those are zygospores. And they're pretty cool looking, actually, under a microscope. They've got these crazy antler structures that kind of suspend them above everything. Pretty neat. So this is the generalized life cycle. Okay. So let's start with a sporangiospore. Okay, so it comes out of the sporangium, it lands and it germinates, growing a single hypha. All right. So if conditions are right, it'll grow this hypha and it develops into a mycelium. Whoops. And then when it's ready to reproduce asexually, it will create new sporangia. Those spores will go out, take over another part of your bread, whatever, or get picked up by a passing insect and maybe make it a kilometer or more. Okay, it can stay in this asexual reproductive circle for a long time. If conditions are right, meaning kind of bad, and it meets up with a compatible another haploid mycelium from the same species, then they can come together, form those little progammetangia, then lay down their cell walls, share the nuclei, and we get our zygospore. Out of the zygospore, well, is it haploid or diploid, this zygospore?
Diploid.
Yeah, it's diploid at first, then it undergoes meiosis, so now we've got haploid nuclei, and it will create a new sporangium right out of it. So that's what grows out of the zygospore in almost every case. There's some exceptions. Sometimes hyphae will grow out, but typically it's a new sporangium, and that's haploid cells again, okay? But they're recombined from the genetics of both parents, okay? Questions about the life cycle?
So the black spots that we'll see on our bread, that is going to be most likely from the asexual reproduction rather than the sexual.
Yeah,
okay.
I'd put money on it. If you put two separate inoculum sources on your bread and they grow together and along the line where they meet, you get really big black dots, then I'd put money on that being zygospores.
Okay, are the asexual spores also called zygospores?
They're called sporangiospores because they were produced in a sporangium.
Okay.
Yeah.
Dr.
So I hope
Tom?
I hope you're seeing the pattern is how things are named. There's like 400 different spore names in fungi, and they're all named after the structure they're produced in. Somebody had a question.
Yeah, sorry. If we were to do this experiment with the two different types from the two different soils, would we have to stress them out in order for them to undergo sexual reproduction, or just putting them together would be enough?
You would have to stress them out.
Okay.
And so that would mean probably just smaller pieces of bread so they run out of food faster. And then maybe take it from like an upstairs drawer where it's warm to a basement to where it gets colder, and they'd be stressed and they'd have sex. Yeah.
Okay. Thank you.
All right. So be familiar with this life cycle and keep in mind that like 99% of their life cycle is spent in this little asexual loop. Okay. If conditions are good, it makes sense to be asexual. It really does. Because if you're doing well in a given environment, your clone is also going to do well. And so it's a way to quickly take over like a slice of bread, right? So you land there, you're growing, this bread is delicious, you're having a nice time, you're growing really well. It's time to make some sporangiospores, which are clones of yourself, and hope that some of them fall, you know, a few inches down onto the other piece of bread, so you can just like rapidly take over everything. This is why, even though you may have different mating types in that pinch of soil, actually it's a good bet that you do, only one of them is going to outcompete the others. Okay, so whoever is better at eating that bread at that temperature with that amount of light, wherever you are, that's the one that's going to take over. And since they reproduce so quickly asexually, they're able to really dominate like a slice of bread or a strawberry or wherever they are. All right, let's take a look at their phylogeny. Okay. They are definitely paraphyletic. They are probably sister to the chytrids, or at least kind of all mixed up in with the chytrids, because the chytrids are also paraphyletic. Zygospores popped up right here. Okay, so it is apparently the ancestral condition of sexual reproduction in the quote-unquote higher fungi, so the non-chytrids. So even our ascomycetes and basidiomycetes have genes, although most of them are degraded or co-opted into doing something else. They do have the same genes as zygomycetes for making these like gametangia coming together, the cell signaling that shows, hey, we're touching now, let's lay down a cell wall. That's all still conserved in basidiomycetes and especially in the ascomycetes to some extent. So we think that zygospores are kind of the ancestral condition for sexual fungi. We've got two main groups. We've got the Mucormycota and the Zoopagomycota. Okay. There are other groups in zygomycetes, but they're either not studied very well or have very few species in them. It's kind of hard to tell. The phylogeny of zygomycetes confuses me quite a bit, and it changes all the time. In your website, I put a link to the ZygoLife page, and so this is a consortium of scientists around the world that work on zygomycetes, and their focus tends to be evolutionary biology, and so you don't find much on that page about what zygomycetes are doing or the genetics of how they adapt to different environments. It tends to be just a bunch of phylogenies constantly updating. And when I started preparing, I was like, I need to get the newest phylogeny of these things, and that was Spatafor et al. 2016 down there in the bottom right. And by the end of summer, it had already been updated, and I just said, screw it. There's no point in keeping up at this point. But these things did remain consistent. They are paraphyletic. They are probably sister to the chytrids, and the zygospore definitely was formed here, or at least the genetics underlying it are found in all of these groups. So it's a nice synapomorphy for them. Okay. Questions about their phylogeny. It's kind of messy. Basidiomycetes and ascomycetes are much cleaner. We have good monophyletic groupings for those, but zygomycetes, historically, they've just been treated as anything that makes a zygospore, and so our phylogenies are still really messy with these. A lot of them also tend to be kind of tough to grow. Okay. So let's look at our two main groups, the Mucormycota and the Zoopagomycota. And we'll start with the Zoopagos. Okay. This is the earliest diverging lineage. These tend to be kind of weird things. All right, so like obligate parasites, insect pathogens. Some of them can only function by living off of other fungi. These are mycoparasites. Okay, and we got three main groups here. The Entomopterales, or Entomopteromycotina, okay, which isn't actually a real group. The Kixellomycotina and the Zoopagomycotina. Okay, say those 3 times fast, right? Entomopteralians, these are insect pathogens, okay? There are a few that are saprobes, which means they break down dead stuff, but for the most part, they tend to be dependent on living insects. And so sometimes you can actually see this. In the summertime, when the flies start getting into the house, every now and then, if you go up to a window, you'll see a fly stuck on the window surrounded by this like puff of white powder. Okay, that's entomophthalmian fungi that have taken over the fly, made it fly towards the light by taking over its muscles, and then killed it from the inside out before sporulating. Okay. We've got, unlike,
how does that fly get stuck to the window? I mean, is it just wait on the window until it dies, or is it just how does that?
It waits on the window, and then it actually the fungus starts excreting like sticky surface from the fly, so it gets super stuck there. In nature, there aren't windows, obviously, and so these actually go to the underside of leaves, thin leaves where the light's kind of shining through, and so they'll kind of glue themselves underneath the leaf, and we don't think that like they evolved to work on windows, right? There has not been windows around long enough for that to have happened, but if they're stuck to the underside of a leaf, then they're protected from the rain, and they can sporulate, and it goes down. And so these flies go up, they stick themselves to the underside of a leaf, and then they die. The biochemistry of how the fungus actually takes over the fly's muscular system is just now being discovered. That Zygolife page actually has some interesting information about that. Jason Steich and his lab group at UC Davis are investigating that. And we used to think that the fungus took over the nervous system of the insect, but it looks like it skips that. So the fly is fully aware, to whatever extent a fly can be aware, of what's happening, and it doesn't have control of its muscles anymore, even though it tries. And so the fly is actively trying to not be stuck to the leaf, and it can't control its muscles, which is really terrifying.
So the fungi actually control the fly to the point where it sticks itself to the underside of a leaf, and then they attach it to the leaf.
Mm-hmm. This is nightmare fodder for me.
So do these fungi, do they end up killing all of the insects that they get on them, or are there some that don't?
They kill them either directly or indirectly. For the most part, it's direct. So like this one, it's actually eating the fly from the inside out after it sticks itself to the leaf. And so it slowly digests it from the inside and definitely kills it. And only when it runs out of food does it sporulate. So the more food it eats, the bigger the fungal body can be, and so the more spores it can make. So it makes sense to eat as much of the fly as possible, and that obviously means death for the fly. There are fungi that grow on cicadas, we just found, that just grow on their abdomen. So they like take over a cicada's butt and then excrete hallucinogenic chemicals, the same things that are in magic mushrooms that people eat. Okay, so they excrete these chemicals, and it makes the cicadas into sex maniacs, and they have sex with everything that moves, which is their abdomen moving up and down, and so that's how the spores spread. So it's like a neat adaptation, if terrifying, to get the spores to spread from insect to insect. Is you make them into a sex maniac, they will spread your spores for you really efficiently. Those don't kill the cicada necessarily, but their butts do fall off, and so they kind of die. But cicadas only have like a 5-day lifespan once they're up in the air anyway.
So can this happen to any insect, or is it mainly just flies?
Oh, all kinds of insects. Yeah,
okay.
Flies are the most studied, but there appears to be a separate species of entomopteran fungi for each species of fly out there. Yeah, so they specialize on one species of fly. This kind of makes sense because they all have their own slightly unique ways of signaling their muscles to move. And so the fungi have kind of co-evolved with the species of flies, and I mean cicadas, all kinds of insects. They
So
tend
theoretically, could they I mean, it could get on like a hornet or a bee or anything like that as well.
Yes. Yeah.
Okay.
They have them for bees also. Bees are pretty good at defending their hive from that, though. Like if they notice fungi growing on a bee and it's acting funny, they'll just kill it and then take it out of the hive, quarantine it, and bury it. And then this is really crazy. The drone workers that take the sick bee away actually stay there and kill themselves. Because otherwise they might be bringing back the fungus with them, you know. And so, yeah, fungi are bad if you're an insect. You really have to worry about them. For us, not so much. And we'll see that when we talk about medical mycology, but our body temperature prevents most fungi from growing. All right. So the Emphthymothorales, really cool fungi. There's apparently quite a large number of species. Some people think that for every insect out there, there's an Emphthymothorine fungus that has co-evolved with it, and we just haven't found it yet. So that would mean millions of species of these things, but we're not sure about that yet. They're kind of hard to study. We've got our Mycology. These are mycoparasites, and some of them are symbionts of insects, so they don't kill the insect. They just live in the insect. The insect kind of spreads them around, and they do their sexual reproduction, their zygospores, outside of the insect. Okay. And then we've got our Zoopagomycota, mycoparasites, predators and parasites of invertebrates. These are the only fungi we know of that specialize in eating amoeba, but they're tough to grow. I can only think of 2 labs in the world that are actively studying these. Just brave souls that wanted to work on something really difficult to study. Okay, so those are our main groups. You can kind of see the weird diversity that goes on. What kind of hyphae do they have? All 3 of these groups, what kind of hyphae?
Aren't all zygomycetes aseptate?
Yep, there you go. It wasn't meant to be a trick question. Yep, so they're aseptate, and they do make zygospores, as seen here. Okay. What we're looking at on the fly's butt is those are not zygospores. What are they?
It's just hyphae?
Nope. It's a type of spore.
Oh, just sporangia?
Yeah. So sporangia and sporangiospores. Yep. So this is asexual on the fly.
Right, because they have a lot of nutrients.
Yeah. All right. Then we got the biggest group of zygomycetes is the Mucormycota. Okay. These are sister to Dicaria, and so they're closely related, more closely related to mushrooms than they are to the Zoopagomycota, for instance. That's just because, you know, these are paraphyletic group. Okay. The Glomeromycotina is another group that's really hard to study because they all look exactly the same and their genomes are humongous, and so it's kind of tougher to work with. But we call these the arbuscular mycorrhizal fungi. 80% of all plants on Earth depend on Glomeromycotina to survive. Okay. Most plants cannot get enough nutrients on their own. They need these fungi. And they're called arbuscular mycorrhizal because they form these little arbuscles that look like a little tree inside of the plant cells. Okay. And mycorrhizal just means a fungus in association with a root. Okay. So this is a plant root. Here's the spore mass of one of these AMF fungi. They grow in between the plant cells, and then they form these little arbustles inside, and that's where they're exchanging nutrients with the plant. Okay, so this is a nice mutualistic arrangement. The plant is getting nitrogen, potassium, good stuff from the fungus. The fungus is getting carbon from the plant. Okay, because plants are really good at taking air and turning it into sugar. This fungus really wants sugar. The fungus is really good at getting nitrogen from the soil, and so it makes a nice little trade-off. These spores are actually formed underground, which is weird. Okay. The mechanisms and reasons for this are still not well known, but a pretty recent study just a few weeks ago did come out and show that these spores are getting around. They did a cool little bit of research where they took sterile soil and planted a green roof on a building on their campus, University of Minnesota. And so we didn't have any fungi in there because the soil had been sterilized. They sterilized all the plant roots that went in, and they planted sterile seeds and everything. And so they did this, waited a year, and then dug up some soil, pulled some roots out of it, and they found arbuscular mycorrhizal fungi there. And so these things are getting around somehow, even though the spores are formed under the ground. How? They don't give an answer. I don't know. It's just happening. They were thinking it could be birds scratching around in the dirt, picking up worms and stuff, and then they fly to the roof and they've carried the spores with them. So these things might be dependent on larger animals moving them around. No idea. All right, so that's our Glomeromycotina. This is a really important group of fungi, but it's not thought to be very species rich. Okay, there may be a few hundred different species that we know of. Could be more, but we're not sure. Okay, and then we've got the Morchiorello-Mycotina. These tend to be saprotrophs, meaning they just eat dead stuff. But there are some of them that live in plant roots. My dog's crying. Hang on a second. Go on, get out of here. Okay, I'm back. And then we've got the Mucormycotina. All right, so this is what the Mucormycota is named after, because these are our most common zygomycetes that we find. Here's the black, sticky-looking blobs, okay, growing on, I believe this is a strawberry here. So if you have a strawberry, let it get really old, it will get black stuff on it. These things tend to be, Mucor just loves strawberries, okay, good sugar source. And strawberries grow really close to the soil, so it's perfect for them. Okay, so Mucormycotina, soil, dung, plant debris, sugar-rich plant parts, they're found all over the world. The number of species here in Mucor and the abundance of them really dwarfs the other groups. And a lot of people, when they think of zygomycetes, they don't think of Enteropteran, like insect parasites. They think of Mucormycotina, which is kind of I understand that. It's a fair opinion. Okay. Those are our two main groups. Any questions about them so far? Nothing? All right.
Actually, I do have a question. Just my
Sure.
microphone's taking a while to click on it.
I'll wait long.
Yeah. So a while ago, years ago, I saw a report on how some bread mold was inhaled by this man, and he ended up losing quite a bit of his nose and face.
Yeah.
Is that actually possible, or is that just kind of something that may have been a spoof, or what?
No, that's real. And we'll see him when we talk about medical mycology. That one, I mean, it's a horrifying case, and when we see pictures, you'll see why. But
I see a picture of him right now.
Yeah, he was actually the first person to have like a full face transplant and survive it. He did later die, but mucormycosis is one of the most common fungal diseases in humans. and it is caused by these things.
So
Now,
was it the bread mold that, like the spores from the bread mold that were able to, he just inhaled it and that's what happened? Or?
yeah, they grew in his mucous membranes and dissolved his face and even bone from the inside out. This is very uncommon. There's a reason you've only heard about that one person. Mucormycosis does actually happen a lot. There are thousands of cases a year, but typically not that bad. Typically, it gets in your lungs and will start growing in there, and you have to take strong antifungals to get rid of it. But I wouldn't live in fear of bread mold if I were you.
Right. Like I said, I saw this a while ago, and I wasn't sure if that was something that actually happens or if it was something that, because we're talking about bread mold and whatnot, so that's why I brought it up.
Yeah. So these are an important human pathogen, but they depend on pretty severe immunocompromised people in order to grow.
That makes sense.
Yeah. So HIV patients have to worry about this. People taking immunosuppressants for like an organ transplant definitely have to worry about this. But most people, it's not going to be a problem. Yeah. Now, that said, it can be an allergen. And so I think it's estimated about 6% of all people on Earth have allergies to Mucormycotina, some of the surface proteins in them. And so that's just going to be like a case of sneeze, runny nose, itchy eyes kind of stuff.
Right. Okay, thanks.
All right. So we're going to take a closer look at some of these structures. Your bread mold is probably going to look like number 1 on here. If you were to manage to pluck one of those balls off from your bread, the little black balls, stick it on your slide and look at it with the scope, kind of crush it down with the cover slip a little bit, I would expect that you would see number 1 right here. That's a Mucormycotina typical spore mass. Okay, it's really sticky and goopy. The stickiness is because they depend on insects to kind of move them around. Okay, so they stick really well to insects. Some of them can actually forcefully eject their spore mass also. But look at the wide variety of sporangia here. It's pretty crazy. This is how people have traditionally gone about describing new species morphologically. It's not the hyphae. All the hyphae look the same. It's a clear tube, looks like spaghetti. Okay, it's identical across all these species, but the reproductive structures can vary quite a bit. Okay. So we've got all these nice, like, well-developed sporangia here. We've got weird little things that just have a few dozen spores on the outside. And down here in the bottom right, numbers 13 and 14, those are actually sporangia also, but they look more like conidia from an ascomycete. Okay. What we think, looking at phylogenies, the best ones we've got, is that the trend is to move towards number 13 and 14 and away from number 1. So less complex sporangia. Right. Here's an electron, scanning electron micrograph. So we've got our sporangium there. You can see right here the nice little septa where it's closed itself off and kind of isolated this part of the fungus. Okay. We call this the columella. That's kind of where the spores are going to start growing. And you can see that they've just laid down little cell walls around nuclei, and we call that a spore. All right. Here's some of those reduced sporangia up close with an electron microscope. You can see the difference here. It's like a lot less well developed. You still have a good septa there, isolating it from the rest, but there's no deep differentiation. We don't have a strong columella where spores are produced on the outside. So we call these marrow sporangia. It's just kind of, I'm not going to require you to know that name because they are sporangia, They just wanted to give them a separate name because they look different. Okay. The evolutionary trend is towards these reduced sporangia. When we look at ascomycetes up close, we'll see that their reproductive structures look more like this. Okay, and so the more derived versions of zygomycetes that have difference from the originals look more like this. All right, here's a close-up of zygospores, so sexual reproduction. In the top left, this is what I'm telling you you might see on your bread if you put down two compatible strains next to each other. See how they just grow all out asexually. When they meet in the middle and run out of food, there's that nice like line of zygospores. So if you zoom in on that, you're going to see all these different stages of zygospore production. Okay, so here are the progammetangia meeting up. Here they are starting to form the little antlers that grow around it. The nuclei are being passed between these at this point. And then here, it's kind of hard to see, it's so bright, but there's a nice ball. That's the zygospore right there. And it's being suspended by these antler-like protrusions. Okay. Anyone give me an idea why it grows antlers? Just best guess.
Surface adhesion?
Yep. These stick to things like bee legs really well, okay? Bird legs, feathers. So it kind of works just like a cocklebur or whatever that gets stuck in your dog fur or on your socks. Okay, it sticks to stuff. And so that's its main adaptation for getting around. Okay. Let's look at a couple of these groups. This is Pylobolus, which means the hat thrower. Okay. These are dung fungi, so they need like partially digested grass basically to grow in, and they have to pass through an herbivore gut in order to germinate. So it's similar to like a tomato seed in that way. They need like an acidic bath. Okay. Here, this is the sporangium, little dark spot on the top, and that's the little hat, basically, thus the name Pylobolus. Okay, here it is growing in some dung. You can see it's got this nice swollen region. It's differentiated. That's got a bunch of spores in it. That's the sporangium. But Pylobolus is famous for being, one, really easy to grow, and two, shooting its spores. So here we see them growing and then just disappearing. So it's swelling up. You can see that it's kind of like laying down that septum. Okay, so we've got some differentiation. Then it throws down some pigmentation to protect it from UV light, and then it disappears into thin air. In slow motion, it looks like that. So it's using water pressure to launch this thing. And so its life cycle actually depends on, I don't know if you know this, but cows don't typically eat their own poop. They eat grass. And so what this wants to have happen is it wants to land on some grass and be eaten by a cow or a deer or whatever. Okay, it passes through the herbivore gut, which activates germination. It grows in the dung. It's ready to reproduce. It's got to get off of the dung in order to be eaten again. And so it launches itself. So it does that by excreting sugars into that, like the bulbous region right there. That makes water rush in because of osmosis. And eventually it gets too much pressure and it just launches. Right now, this is the fastest or the highest acceleration known in biology is this particular species, 20,000 Gs, which is crazy. These are really light sensitive, and they have a cool little mechanism. You can see here, like, this is showing the path of light going through here. They're directed towards the light. That bulbous shape there, that's kind of clear, you can see that. It's actually refracting the light. It's got a higher refractive index in the air, and so it's focusing all the light right on the tip there, and that keeps this thing pointed towards the direction of the sun at all times. And since they grow really fast, we're talking like in a few minutes it can change the direction that it's growing. It grows fast enough towards light that in a dark lab you can actually hold a flashlight, and if you're patient you can get this thing to make little shapes for you. I spent a while trying to get it to make my initials, but always gave up. All right, so that's kind of a weird zygomycete, Pilobolus. You'll see it in your book, but it's one of those random Mucormycotina that do some really crazy stuff. All right. Commercially, zygomycetes are really important. That's how we get sake, the Japanese liquor. That's how we get tempeh, the Indonesian delicious soybean food. If you are interested in making your own tempeh, I have Rhizopus oligosporus cultures, and I can take those into the lab for you. Basically, you just cook some soybeans, let them cool down to about body temperature, and then throw some spores on it, and you get tempeh. Okay. So let me know if you want me to bring some of that in, and I'll take it to the office. You can pick it up. All right, so here's what you should be able to figure out at this point. I'm gonna give you a minute to look these over. Are we past time?
No, we have like 25 minutes almost.
Oh, okay, okay. So read through these, see if you can figure it out off the top of your head. If not, ask me about it. Not all of these will be immediately apparent from our lecture. You may have to do a bit of the reading also if you've done any of that yet. All right, who can take question number 1 for me?
They would be haploid, right?
Yep, necessarily haploid in every case. All right, question number 2.
Okay. So I'm going to say derived because we said that zygospores inherited that trait from a common ancestor.
Yep.
So that means that not having zygospores means that they had them at one time and then they evolved to not have them later.
Yes. As a matter of fact, the fact that they didn't have zygospores meant that for the longest time, like up until 10 years ago, we didn't consider them part of Zygomycota at all. Glomeromycota actually had their own phylum because they were unlike anything else. And they have really huge genomes and it's tough to work with and all that. And so we just recently found out that they do fit in with monophyletically with Mucormycota. Yep. They just don't make zygospores anymore. All right, number 3.
Okay, I guess I'll go again. What I would think if something starts off as only asexual and then starts having sexual reproduction means that it's either having a lot of pressure on it or it's starting to branch out into different environments than where the asexual reproduction mode was working.
Let me correct one of your assumptions about this question you might have missed. The conidia, the merosporangia, are also asexual.
Okay, never mind then.
So this is saying merosporangia have evolved from sporangia. What's that say about sporangia? The evolutionary pressures on it.
What is this referring back to the picture of the sporangia where it said everything was moving towards 13 and 14?
Yeah. So it seems pretty clear, even though our phylogenies aren't perfect and they're kind of shifting around and some uncertain stuff is still in there, there is a clear trend towards reduced sporangia. So for whatever reason, the environment is selecting over time, or has been selecting over time, I don't know if it still is, for a simpler spore device, I guess. It takes fewer resources and you still get your spores out there into the world. Okay, number 4, Pilobolus, the thing we just saw shooting its spores.
This is the one I had a question about, so I guess I'll try to answer it. But
Okay.
I'm gonna guess. I'm just gonna guess. I have no idea, but I'm gonna guess that it is a zygospore, and that it's a diploid. So that way it goes through. Then after that, it can go through meiosis and then grow sporangia and spread in the gut, I assume.
I hate to inform you that your guess was wrong.
Oh, exactly.
Okay.
I tried.
So if it's not a zygospore, then it's a
Spore, well, gametospore, whatever we call. No.
sporangiospore.
Sporangiospore. Thank you. I can't remember the word.
Yeah, so that actually is a sporangium. You'll find the sexual mating types right around fall, as the weather gets colder, and they'll be on the dung, and then they'll make a new sporangium that can actually overwinter, and so they wait for the grass to come back, essentially.
Okay. Cool.
The zygospores are pretty good at waiting over winter. The cell wall is a lot thicker than a sporangiospore, and so they can just hang out for months. You can freeze them; it's no problem.
Yeah. Well, I'm glad.
And so, are they haploid, diploid, or dikaryotic?
Haploid in that case.
Haploid. Yep. Okay, who wants to explain why filamentous growth is so common across all microbes, including bacteria?
I would basically just say it's an effective way of absorbing nutrients from the environment.
Yeah, it's pretty efficient. Okay. If you're a lysotroph, you can be one of 3 shapes. You can be a tiny blob, you can be a tube like humans, or you can be mycelial, filamentous. Those are the only 3 that actually make sense, and they're the only 3 that we find. This last one, we're gonna have to wait for the reading, so keep that one in mind. The Spitzenkerper, okay? And then, all right, last question. Difference between homothallic and heterothallic.
Okay, so the homothallic, if I remember the terms correctly, was the few species that are able to make zygospores on their own,
Yep.
And then the hetero needed another individual.
Yeah. So homothallic can have sex with themselves, heterothallic can't. Yep. All right, so these are the kind of questions to keep in mind as we move forward, and they'll be similar when we see our final two groups of fungi also. I want you to fill this in on your own. How do zygomycetes fit into our overall picture of fungal evolution, morphology, physiology, diversity, and ecology? What are the kinds of things that they do? When and where did they evolve? Where do they break the rules of fungal morphology? Where do they conform to them? And then the physiology, some of that's going to come from your chapter, and some is going to come from the paper that you read, the Fisher et al. 2018. And then what kind of diversity do we see in terms of how they make a living, where they live, what they eat, that kind of thing. All right. So your assignments this week. Do your reading. Okay. You've got your Canvas quiz. It is available now, and it's due before class on Thursday. Then we'll have our discussion about the readings. If you haven't yet, go ahead and see if you can get a photograph of your bred zygomycetes. Look for sporangia. If you can find a picture of that, that's awesome. But at least show me the hyphae on there. And go ahead and, I didn't write this here, but go ahead and isolate some of that on one of your agar dishes, because we're building our fungal culture collection. All right, and then finally, I'll stop sharing my screen here. Okay. So finally, what I want you to do is look in your little lab kit that I gave you. Inside the big Petri dish is a tiny little tube labeled pollen. Okay. What do I want you to do with that?
Put some dirty water in there.
Yeah, add some water. It doesn't have to look dirty. It doesn't have to be like brown or whatever, but you know, go to a lake or a little stream or a pool of water next to your house or whatever, a kiddie pool, whatever it is. Ideally not chlorinated, okay? Find some water, just drip some in there. Loosen the cap a little bit so we can get some oxygen exchange. We don't want it to go anaerobic. And then wait a week. Okay.
So we just need like 2 or 3 drops.
Yeah, just a few drops. If you want to fill it like halfway, that's fine. Then just kind of shake it up a little bit lightly. You don't want to shake it too much. And then loosen the cap, leave it upright somewhere for between 3 and 7 days, and you'll get Chytridiomycota on there. And we should get some chytrids with little rhizomorphs growing, and you'll see a little ball hanging out on your pollen. So to visualize that in your scope, just take a little bit of that water and drop it onto the slide, just a tiny piece. Make sure that some pollen kind of gets in there. Put a cover slip down and take a look at the pollen, and you should start seeing that after a few days. Okay, so get a photo of that also. I hate to inform you that there's no easy way to like keep a fungal culture collection of chytrids. They require that pollen to live, or like a frog or something. So don't worry about trying to put that one on a plate. It won't grow. It won't survive. Okay. Any questions?
Yeah. So
Okay.
You want a photo of our bread mold for Thursday. If I don't have anything, do you want me to just send in a picture of the bread that I'm trying to grow it on, or?
No, you can wait.
Okay.
These deadlines aren't hard coded. I know that fungi aren't going to behave the way we want them to all the time, so that's all right.
Okay.
Get it when you get it.
Am I
The
good with
true due date for all these assignments is going to be the end of the semester. Obviously, it's better if you just keep up with it and get it out of the way as soon as you find the stuff. But yeah.
I might do the same thing because I have too much green stuff growing.
Yeah.
And I got it from the surface as well, so I'm thinking that's probably the problem.
Yeah, maybe dig a few centimeters down.
Oh, deeper, yeah.
Any other questions? All right. Well, I will see you on Thursday. Don't forget about the reading and the reading quiz, and we'll chat about it on Thursday. And then next week we will start exploring the ascomycetes. All right.
Thank you. Have a great day.
See you guys later.